ESF had been on my radar. Then it wasn't for a while until the water cluster hire popped up, and my mentor sent it to me and said, oh, you should, consider applying to this. And then it was like, oh yeah, ESF. What I really like about ESF is that we do have this small school community feel. It's provided such a great sense of community as I embark on new faculty life, and facilitated many collaborations and friendships within ESF. I started teaching right away when I moved here. I was doing HR paperwork one week, and in ILIC 5 the next, teaching cell biology, which is one of our core courses in the environmental biology major. As a lecture course, we cover the types of cells, how we study them, how they respond to stress and grow, and was particularly proud of the evolution of cell this past year, and making it more my own, and honing in on the major questions that we focus on throughout the semester. How genetic information is transferred through the cell, like how cells get energy, things like cell communication, things like that. And then also implementing our final project themed on model organisms and the study of cell biology. It was a semester-long project where students worked in small groups and read papers on what we call an emerging model organism, is something where there's a lot of dedicated study of a certain aspect of this organism, cell biology. And we called the whole endeavor SUNY ESF's next top model organism. The students, instead of having a final exam, had a poster symposium where they shared these results with each other and faculty and graduate students, and it really brought the course to life. I also co-taught tropical marine ecology with Josh Drew, and that's a sequel course to Dr. Drew's Corals Conservation and Colonialism course. During the spring semester, we prepare for spring break trip to Vieques, Puerto Rico, where we went on a treasure hunt for bioluminescent dinoflagellates that create the glow of the world-famous bioluminescent bays. This is what's considered like the brightest bay in the world. And it was really neat that we got to do science there. So we went to the bioluminescent bay and some other nearby ecosystems to look at the plankton and the water column, and also the sediments, and hopefully try and characterize how these populations change through time as we return to Vieques with future iterations of the course. Broadly, my research group studies microalgae and their interactions with host partners, whether they be bacterial symbionts, coral animal hosts, or freshwater sponges. Those are just microalgae interacting with their environment really excites my group and I. A research area that we're active in right now is microalgal bacteria interactions and how they pertain to coral reef systems. We're also working with freshwater sponges a bit and seeing if we can characterize the diversity of host, or the animal sponge and the algal symbiont partner pairings around New York State, because freshwater sponges are important for water filtration, ecosystem health, and the ability of different host symbiont pairings to thrive across New York State ecosystems. Might be a neat water quality indicator, so that's something that we have going on collaboratively with some of the labs in the chemistry department. When I tell my colleagues outside of ESF what ESF students are like, I first tell them the anecdotes of, these are students who, after it rains, they take earthworms off the sidewalks and put them back in the grass. ESF students really do give us all hope, I think, and that unbridled passion for studying the environment and making the world a better place, and that focus and drive. ESF being the unique learning environment that it is, it really channels a unique type of collective empathy and drive for learning about the world and then making it a better place that really I haven't seen anywhere else.